Bismillahirrahmanirrahim. This lecture regarding the third week of development and formation of trilaminar germ disc. The objectives of this lecture are to describe the formation of primitive streak and primitive node, describe the formation of three layers, ectoderm, mesoderm, and endoderm, describe the formation of notochord, and identify the subdivisions of secondary mesoderm, and of course to define new terms. Starting with gastrulation, and gastrulation mean or defined as the process that establishes all three germ layers, ectoderm, mesoderm, and endoderm in the embryo. Also, beginning of formation of primitive streak on the surface of epiplast at the same time of formation or enhancement of formation of these three germ layers. Primitive streak is a narrow groove in the epiplast, slightly plugging region on either side. Primitive node is a slightly elevation in the area surrounding a small primitive pit. Here, this figure of an embryo in third week. Here we have the amniotic cavity and yolk sac, and here we have the embryonic disc with epiplast and hypoplast, and here this is the area of connecting stalk. This is extra embryonic mesoderm, cytotrophoblast, and syncytiotrophoblast. And this section from this area showing here cut edge of amnion and cut edge of the wall of yolk sac, and here we can see in blue epiplast and in yellow color hypoplast. And here is a buccopharyngeal or oropharyngeal membrane, and this is a primitive streak, and invagination in the epiplastic layer. Also, this figure showing cut edge of amnion, and here is the primitive streak, and here is the primitive node. Okay, the cells of the epiplast migrate toward the primitive streak, and then it has an inward movement, and this inward movement of epiplastic migrant cells is called invagination. The invagination is the process by which cells migrate from epiplast inward movement through the primitive streak. So some of cells displace the hypoplast. Here the hypoplastic layer displaced by migrating cells from epiplast, and this form the endoderm. Now, other cells come lie between the epiplast and newly formed endoderm to form the mesoderm, while the cells that still remaining in the epiplast form the ectoderm. Hence, we have to notice that all germ cells sorry all germ cells, ectoderm, endoderm, and mesoderm, form it from the epiplast. And this figure showing the movement of these cells or migration of these cells from the epiplast through the primitive streak. Firstly, they replace the hypoplast to form the endoderm. After formation of endoderm here, other cells migrate and lie between the endoderm and the epiplast, and this form the mesoderm. Here is the direction of migration from epiblast anteriorly and laterally. Here we have the buccopharyngeal membrane and cloacal membrane. That means we have here the head side and tail side. So the primitive streak appear or start from the just near the cloacal membrane and moving cranially. The secondary or intermediate or intraembryonic mesoderm, we have extraembryonic mesoderm in the chorion, if you remember the last lectures. Here we have the secondary mesoderm or intraembryonic that form from the epiplastic cells that migrate through the primitive streak and lie between the endoderm and epiplastic cells. Okay, from ectodermal cells that side the primitive groove, and here the spread or migration of the cells occur in all directions of the embryonic disc, but we have three regions still remain in two layers, not three layers there. Only we have ectoderm and endoderm, and these layers or these regions are oropharyngeal membrane, notochord, and cloacal membrane. Starting with the notochord is a cylindrical rod-shaped structure induced by primitive node area. Some genes induce the formation of notochord. It lies in the midline between the ectoderm and the endoderm. The extension of the notochord from the primitive streak to precaudal plate. The fate of notochord later on, it forms the basis of vertebral column and induce the overlying ectoderm to form the neuroectoderm and neural plate. This figure showing here, we have this section sagittally cutted to show the primitive bit here, and here we have the epiblast, and here the notochord, starting from the primitive bit and forward. So this diagram also showing the formation of the notochord from plate. It's called notochordial plate, and then it's turned from side to side to form the notochord finally. After formation, it's released from epiplastic cells and endoderm in this region. Just lateral to notochord, here the mesoderm in this region arranged as paraaxial mesoderm. Regarding the formation of the notochord, we have pre-notochordal cells that invaginating in the primitive pit. These cells move forward toward the head or in cephalic direction till they reach the precaudal plate, and these cells become intercalated and form two cells layer that form notochordal plate, and this plate proliferate and detach from endoderm, and finally they form a solid cord cells. This is definitive notochord. And here is a primitive streak, precaudal plate, and here we have the area of buccopharyngeal membrane. This is a caudal end, and here this is a primitive node, and here we have notochordal process. And as I mentioned before, the notochord itself has the formation of neural plate. This is the neural plate, and here is the neural plate expanded, and here is the notochord overlaid by the neural Here, this figure around the 15th day, 2 weeks almost, 17th day, 18 days, and here after 21 days. So the caudal process grow forward from the primitive node as I mentioned between ectoderm and endoderm, as well as it reaches the precordial plate or the area of buccopharyngeal membrane. So the notochord underlies the neural tube and serves as the basis of the axial skeleton. Also, notochord and pre-notochordal cells extend cranially to precordial plate and caudally to the primitive pit, and this is the extension of the notochord. So the pit form as indentation in the epiplast for neuroenteric canal. This later form the ventricles and canals of the brain and spinal cord. The cloacal membrane is formed in the caudal end of the embryonic disc, and it's appear on the posterior wall of the yolk sac and form a small diverticulum extend into the connecting stalk. And this diverticulum called allantoenteric diverticulum or allantois. What is the fate of notochord? The notochord itself, it leaves, but uh, some remnant appear after disappearing of notochord, which are including the nucleus pulposus of interventricular discs. The disc between vertebrae is called intervertebral disc. It has outer fibrous and inner slightly semi-watery in the center. It's called nucleus bulbosus. The nucleus bulbosus formed by the notochord. Also, apical ligament that connect the odontoid process of axis and occipital bone formed by notochord or remnant of notochord. Sometimes abnormal presence or persistence of notochord give rise to malignant tumors called chordoma. Regarding the gastrulation, we have the fate map for establishment during gastrulation. Firstly, the cells that ingress through the cranial region of the node become notochord, and cells that migrate to the lateral edge of notochord become paraaxial mesoderm, and cells that migrate through the midstrict region become intermediate mesoderm, while the cells that migrate through the caudal part of strict form the lateral plate mesoderm. Hence, we have here the three types of intraembryonic mesoderm. We have paraaxial mesoderm just lateral to the notochord, an intermediate mesoderm lateral to paraaxial mesoderm, and finally lateral plate mesoderm with most lateral layer of the mesoderm. This figure showing the uh, parts of mesoderm. Here we have the notochord. Just lateral to the notochord we have paraaxial mesoderm. And lateral to and caudally to the paraaxial mesoderm we have intermediate mesoderm. And lateral to intermediate mesoderm we have the lateral plate mesoderm there. Paraaxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. Here we have the cloacal membrane and buccopharyngeal membrane. Head end and tail end. Regarding teratogenesis associated with gastrulation, that abnormal accumulation of tissues, it's begin at the third week of development, which high sensitive stage for teratogenic insult. Example for these teratogens: high dose of alcohol kills the cells of anterior midline of germ cells. So the result of this teratogenic factor is holoprosencephaly. You can see also this case in the next slide. Gastrulation itself may be disturbed by genetic abnormal and toxic insult. Any genetic problem or toxic insult, it may cause caudal dysgenesis. That means the caudal part of the embryo have difficulty to grow or to develop because of insufficient mesoderm in this region or caudal part. It may cause caudal dysgenesis or Sirenomelia. Also, situs inversus, and this is a condition transposition of viscera of thorax and abdomen may be occurs. That means the right being in the left side and the left side become in the right side in case of situs inversus. Also, other conditions of abnormal sideness may be known as laterally sequences. In this case, patients with this condition do not have complete situs symphysis but appear to be predominantly bilateral left-sided or right-sided. That means structures move to one side rather than being in same sides. Here is a case of holoprosencephaly with hypothyroidism and absence of nose. Also, this is case of Sirenomelia or caudal dysgenesis caused by faulty gastrulation. Okay. Also, we have tumors associated with gastrulation. Sometimes primitive streak remnant persists as a sacrococcygeal region and it's called sacrococcygeal teratoma. Also, this tumor may be arise from primordial germ cells that fail to migrate to the gonadal ridge or gonadal area. If you remember from lecture of gametogenesis, primordial germ cells migrate from yolk sac to primitive gonad or gonadal area or gonadal ridge. Failure of migration also may be result in tumor as well as association with gastrulation or abnormality in gastrulation in this topic. Here, the sacrococcygeal teratoma. In this case, arise from persisting primitive streak. The females more affected than males. Also attached in the sacral region, may be containing bones, cartilage, hair, nerve tissue, or other tissues. Disturbances of gastrulation may be summarized as a vectoral word. It may be lead to vertebral defect or anal atresia or cardiovascular anomalies or tracheoesophageal vestulas or renal abnormalities and limb defect. We can summarize all these defects may associated with gastrulation by the word vectoral. Again, this vertebral defect, anal atresia, cardiovascular anomalies, tracheoesophageal vestula, renal anomalies, and limb defects. We talk about the imperioblast. While the trophoblast, as I mentioned in the last lecture, we have three forms of villi. Primary villi, which appear at the third week of or a beginning of third week or at the end of second week. Secondary villi appear after by mesodermal penetration of the sinus or the villus, while by the end of the third week we have the tertiary villus or definitive placental villus. This figure may show the difference between the sets of villi: primary villi, secondary villi, and tertiary villi. Here, the primary villi with the core of cytotrophoblast, and peripherally we have syncytial trophoblast. Secondary, we have the mesenchymal core arrived from extraembryonic mesoderm or mesenchyme, surrounded by cytotrophoblast and syncytial trophoblast. Formation of capillaries inside the mesenchymal core transform the secondary to tertiary villi. So the presence of blood capillaries in the villi, the sign of formation of definitive placental villi. The cytotrophoblastic cells villi penetrate progressively into the overlying syncytium until reach the maternal endometrium and forming outer cytotrophoblastic shell. That villi extended from chorionic plate or decidua basalis, the part of endometrium, decidua basalis. We have a lot of types of decidua. One of them is decidua basalis, where the placenta will form is called stem or anchoring villi. Between the 19th to 20 day of embryo of intrauterine life, the embryo attached to trophoblastic shell by narrow connecting stalk, as we mentioned in the last lecture. And this connecting stalk later on develop into umbilical cord. In summary, the third week we have formation of 3 layers of germ disc, and it's called trilaminar germ disc. Formation of notochord and primitive streak also started. Gastrulation, and as I mentioned before, it's a process by which bilaminar disc converted or transferred into trilaminar embryonic disc. The primitive streak appear at the 15 days as a narrow median groove formed by ectodermal cells. This figure shows the formation of 3 layers from invagination of ectodermal cells or epiplastic cells through the primitive streak and primitive groove to form the endodermal layer and mesodermal layer. And the mesoderm arranged into paraxial mesoderm after formation of notochord, and intermediate mesoderm and lateral plate mesoderm. Finally, we have 3 formed layers: ectoderm, which the remnant cells in the epiplast; endoderm, formed by migration of epiplastic layer and replacement of the older hypoplast; and mesoderm, which is formed by migration of epiplastic cells between the newly formed endoderm and epiplastic cells. This mind mapping figure showing the fate of cells and area from the cleavage. Cleavage, if you remember, the onset of the continuous division of the zygote from 2 cells up to more than 96 cells. So after cleavage, the cells arrange into outer cells that surrounding the blastocele, the cavity of the blastocyst called blastocele, and these cells line the blastocele called trophoblast, while other cells accumulate and forming inner cell mass. The inner cell mass later give rise to all embryonic layers and cells. Trophoblast, which share information of placenta. The trophoblast give rise to cytotrophoblast, and the cytotrophoblast give rise to syncytial trophoblast, while the inner cell mass differentiated into epiblast and hypoblast. The hypoplast give rise to extraembryonic mesoderm and yolk sac endoderm, while the epiplast give the true germ cells and all tissues and cells of the body. It give rise to amniotic ectoderm or amnioplast and embryonic epiplast. And the embryonic epiplast give rise to embryonic ectoderm and embryonic mesoderm and embryonic endoderm. So the primitive streak appear in the embryonic epiplast and give rise to extraembryonic mesoderm, embryonic mesoderm, and notochordal process. While the remaining part of embryonic epiplast not migrated by invagination through the primitive streak and primitive groove is called embryonic ectoderm, and it also gives rise to neural crest cells. The end of the lecture. If any questions, don't hesitate to ask me or send me your questions, and guys, only via Telegram. I'm going to answer you as soon as possible. So thank you and good luck.